All right, I'm going to go ahead and get us started. Welcome everyone to the Fermentation Association Science and Health webinar, Development of Pickling Technologies and Products. I'm Amelia Nielsen-Stoll, editor of TFA. We are a trade group that was launched to support producers who use fermentation to create delicious and often healthful food and beverages. Our goals are to help educate consumers about fermentation and its benefits, support scientific research into those health benefits, and work with food safety authorities to establish clearer and more appropriate regulations in regards to fermentation. Today, we bring you two great speakers, Dr. Ilenys Pérez-Díaz, a microbiologist with the USDA, and Dr. Ben Wolfe, Associate Professor of Biology at Tufts University. We have many great questions already submitted and reviewed with our speakers. If there are additional topics you'd like to see addressed, please enter them in the chat below and we will try to get to them. I am going to go ahead and turn it over to Ilenys and Ben.
Thank you so much, Amelia. And it's a wonderful and incredible pleasure to have Ilenys with us today to talk about the important research that she's doing in pickling technologies. And what I love about the research that she's doing is, pickling and fermentation are these ancient traditional technologies that people have been using for hundreds and hundreds of years. And she's really thinking about ways that we can advance those technologies using all the amazing sequencing and all the modern microbiology that we have today. Ilenys grew up in Puerto Rico, where she attended high school, and then she went on to do a Bachelor's of Science at the University of Puerto Rico. She then went on to do her PhD at the University of Wisconsin-Madison, where she did some really pioneering work on lactic acid bacteria, those important bacteria that do so much of the fermentation that we enjoy. She then went on to do a postdoc at the United States Department of Agriculture Food Science Research Unit in Raleigh, North Carolina, where she is based today as a scientist and also affiliated with North Carolina State University, where she's a professor and is engaged with activities there. And today we're going to be listening to a presentation first, and then we'll have a discussion afterwards based on the questions that you submitted and maybe other questions that come up in the chat as well. So thank you so much, Ilenys, for joining us, and I'm really excited to chat with you today.
Thank you so much for that introduction, Ben. I am very excited to share with this international audience today. And because of the international nature of our audience, I'd like to start by describing a little bit what the USDA does in the United States. And then I'll share some a description of the projects that we are currently running in the lab. I do have a presentation prepared for you all today that I will share in a minute. And I hope, there we go. So first of all, here you have my email address in case you have questions at the end of the presentation that we may not be able to answer. Also, if you have questions on topics that I will not be touching on today, feel free to contact me at that address, which is here, Ilenys, this is IL, ilenys.perezdiaz@usda.gov. So first of all, what is the United States Department of Agriculture? The USDA Department of Agriculture seeks to find solutions for a number of agricultural challenges in the nation and globally. The agency ran 660 research projects in 2019 that were pioneered or organized, led by 8,000 employees, of which 2,000 are scientists just like me. The efforts in 2019 were spread across 90 research locations, which are mostly in the United States, but also there are a few locations abroad. Our agency operates on a $1.4 billion budget annually, and it has a tremendous amount of opportunities for collaborations with foreign processors. In 2019 alone, we had more than 1,500 collaborative projects that were run in the agency. Last year, we had 3,816 peer-reviewed journal articles produced and 85 new patent applications. We materialized 65 new patents and 51 new licenses. So this gives you an idea of what the mission of the United States Department of Agriculture, specifically the Agricultural Research Service, has in the country and globally. I am assigned to the Food Science and Market Quality and Handling Research Unit, and it's our mission to develop new and enhanced technologies in support of the processed vegetable industry in the United States. It is our goal to develop these new technologies considering the reduction of the environmental footprint, reduction in the use of sodium chloride, in the use of energy, the use of water, the use of preservatives, and the reduction in food waste. We also aim at increasing the safety, the quality, and the nutrition in the new products. So this gives you an idea of the scope of the research we are currently doing and what our goals are. My program specifically, since I've been trained as a research microbiologist, focuses in the use of microbiology and bioinformatic tools to better understand the habitat that permeates in fresh and fermented vegetables. And we use this knowledge to then improve a number of processing technologies for fermented and acidified vegetables. I will be sharing some of the projects with you today, but before we get there, I'd like to share with you the details of the process that is in my mind when I'm thinking about advancing the science of pickling. I spend a lot of time thinking about the commercial cucumber fermentation system, which is a little bit different from the household scale or the smaller artisan fermentation systems, primarily in terms of volume and equipment that's needed. But the systems that I think the most about, it has to do with the fermentation of cucumbers commercially, where fresh cucumbers are collected in giant cranes that then move large volumes of cucumbers into open top fiberglass tanks. And these tanks are generally about 12 feet tall and 10 feet in diameter. So these are giant tanks. A facility can have anywhere between 100 to 1,000 tanks. And once the cucumbers are deposited in these tanks, they are covered, or wood cover boards are positioned at the top of these cucumbers and secured before adding the fermentation cover brine. This is done because the salt content in the cover brine changes the density of the liquid phase and causes the cucumbers to float. So we need those cover boards to retain the cucumbers under the level of the liquid. At that point, the fermentation can proceed in about 21 to 30 days, depending on the ambient temperature. Remember that this is a field process. Another process I think a lot when designing projects is the acidification process, which is different from the fermentation process in the sense that it happens in a shorter period of time. So in the acidification process, fresh cucumbers are put into glass jars, and a cover brine goes into the jars with a lot of salt and acid, high concentrations of salt and acid. To preserve or microbially stabilize these cucumbers, they are then pasteurized and maintained in storage until they reach the grocery stores. And the acidified cucumber products, or better called as fresh pack, are as stable as fermented cucumbers. and they can be found on the shelves at room temperature in grocery stores. So these are the two systems that I consider when thinking about improvements or changes. And this slide here gives you an overview of the projects that we have conducted over the years. We have looked at reducing salt as well as preservatives to increase the sustainability of the systems. We have studied the microbiology of the fermentations, mostly as it relates to the development of started cultures and probiotic pickles. We have also looked at ways to reclaim defective or surplus vegetables and convert them into fermented products that can be used in a number of applications. We have looked at reclaiming the fermentation cover brines that are generated in tank yards. And we have looked at ways to reduce carbon dioxide production during the fermentation process so that we can increase the quality and reduce bloater defect. So this is in a nutshell what we have been concentrated on in the past decade and a half. And I will share 4 main components of the research program as it stands today. The first component has to do with the development of novel technologies, as you have heard already in this presentation. The first technology has to do with the elimination of sodium chloride from fermentations. This project was run in response to industry needs to reduce the levels of chloride in wastewaters. At that point, it was an urgent need that was brought to us from the industry, and we accelerated the development of a sodium chloride-free fermentation that was transferred to industrial production around 2010 and prior to optimization. So we continue to work in this system. We continue to optimize the system. For those of you who like to use your devices and cell phones, you can do a Google search for "Can Science Create a Greener Pickle?" and find a short video on this technology that was created by National Geographic to understand a little bit better what the technology is about. The second project I'd like to talk about today or share with you today was focused in taking the fermentation covered brines that typically look like what I'm showing you in this picture in the bottom left. They're very turbid after they go through the fermentation, but they still contain high amounts of sodium chloride. They contain the lactic acid that's produced in the fermentation, and they become turbid. The chemical oxygen demand is very high in these liquids. And so it was the aim to clarify these brines to potentially reuse them in a fermentation without having issues of softening and so forth. And as we developed the project and got to this stage, collaborations with Dr. Susanne Johannismeyer, who you will be hearing from in a couple of weeks, led us to then do a charcoal filtration step that then eliminates the colors and most of the odors that are present in these fermentation brines, creating this clear brine that then could go back into finished products or back into a fermentation and not necessarily to impact the environment. This project is in its infancy, and we hope to continue developing it for the industry. And then the third project that's related to the fermentation tank yard that I'd like to share with you is related to the enhancement of winterization on these giant tanks that are outdoors. This was developed in response again to industry needs. They were worried that if they were going to eliminate the sodium chloride from the fermentation, that they couldn't use it as an anti-freezing agent to prevent the formation of ice inside these tanks during the winter. And that was of concern because what happens in these tanks is that there is a layer of ice that's formed around, and on top of the tanks. And so it becomes quite difficult to remove these fermented cucumbers from the tanks during the winter when you have a layer of ice on top that could be up to 10 inches in thickness. So that will force them to even sometimes use jackhammers to break through that ice and be able to remove the cucumbers from the tanks. So in this project, what we had done is we instrumented commercial tanks to study the fluctuations in temperature in response to changes in the climate or the ambient temperature. And then we developed some models that help us understand the flow of the heat in the system and understand what kind of added insulation was needed. One of the main lessons learned from that modeling is that we absolutely needed to cover the top of these tanks because there was a lot of heat that was lost through the top of the tanks. So we decided to design a couple of prototypes. We ended up testing 3 of these prototypes in the Midwest and here in North Carolina to then maintain or explore maintaining the inner temperature of these tanks above the freezing level, and also maintain that temperature at least 10 degrees above the ambient temperature to enable a fermentation during the end of the spring in locations like North Carolina, where the temperatures are still a little bit low for a fermentation, but if we keep it higher, we can enable the start of a fermentation season a little bit earlier. So those were the goals there. We had made progress. We still need to better understand how to instrument these tanks so that we can prevent freezing when we have temperatures below the freezing point for extended periods of time. So we were successful in preventing the freezing when the cold temperatures occur for short periods of time, but not for long periods of time. So that will be the next step here. And then changing gears a little bit here, I'm focusing on acidified cucumbers. And in the United States, we unfortunately cannot afford to collect small cucumbers, manually harvest small cucumbers from the field. So products that require small cucumbers in the United States then are associated with the importation of the small fruits from overseas. And these, in order to achieve that transportation from overseas, these small cucumbers have to be microbially stable. And that's achieved by adding high amounts of acid, salt, and sometimes sulfite preservatives in the system, in barrels, which ends up potentially polluting the fresh waters here in the US. So what we had done here was look at alternative ways to preserve these fresh cucumbers for transportation from overseas. We were successful in reducing salt and acid content. And then as far as preservatives, we looked at a number of active ingredients that are found in spices such as dill, cinnamon, and mustard seed, because they enjoy greater acceptance from consumers. So we wanted to incorporate those ingredients in the acidification treatments as a way to maintain microbial stability during transport. We were successful in identifying at least 3 treatments that are appropriate for transporting cucumbers from overseas. We tested those at the semi-commercial scale, and we look forward for the adoption of these treatments by commercial processors at this point. So with that, I'm wrapping up the first component of this presentation out of 3. The second component has to do with the reduction of food waste and the creation of foods for a growing population. Sadly, we now know that in the United States, we are losing between 30 and 70% of the fresh vegetable production. So those are alarming numbers, and even though they are estimates, it is necessary to look at ways to resolve the impact of such waste. And so what we are doing there is to develop fermentation systems that are smaller in size, so that they can be used in the farm setting, farmer markets, or restaurants, grocery stores. Perhaps this will in the end be materialized as in a cooperative model where all the surplus vegetables are centralized at a location and then repurposed. In any case, the end goal here is to develop a system that is smaller in size, that is more portable, more manageable by smaller processors, and that can then be used to ferment or acidify a variety of vegetables, with the intention that then we can convert what it was once waste or surplus or a defective vegetable into a value-added product. It could be a probiotic product, it could be fermented vegetables or slurries that then can be used as sources of flavor, colors, ingredients in a number of recipes, or even dehydrate them for different applications. So that's the vision here. We are in the final stage of testing this small volume fermentation vessel for cucumbers. We were almost wrapping up this project before the pandemic, and now it's been 6 months since we've been able to go back. But it will probably be ready for transfer to processors within the next year or so. Along those lines, we are also learning how to appropriately ferment and acidify other types of vegetables, not just cucumbers, because that's where we can better impact the reduction in waste. So we have started to look at how to appropriately brine a number of vegetables, like tomatoes in this case. And I'd like to explore this case a little bit further, because I also want to communicate to the audience the necessity of doing these processes right. This fermentation is a very simple technology. Acidification is a very simple technology, but they need to be applied appropriately with the purpose of preserving the safety of these products. So, for example, in the case of tomato, it has a very waxy skin or peel. And so that is a major consideration in determining how we want to process this vegetable. In this case, we had tomatoes that had an initial pH around 4.6. And that's really the cutoff value to prevent sporulation of Clostridium botulinum in a closed jar, and to prevent the formation of the botulinum toxin that can be very, very damaging to human, even in small concentrations. So here we have to be very careful. Once we identify this parameter, it becomes critical to keep that pH well below 4.6. And that we understand we are brining these these tomatoes appropriately. In this case, we brine these tomatoes in a jar, and we looked at the pH of the tomato itself after 24 hours and 48 hours, 1 and 2 days. And we were finding that the pH was not equilibrated with the brine. It was after 48 hours, it was at 4.28. There was some change in the right direction, but the system was not equilibrated with the brine. And so the goal is to be able to equilibrate the brine pH with the vegetable pH, so that we can achieve that long-term microbial stability. We did not achieve that with the tomatoes. And then we tried a number of things in terms of the shape of the tomato we wanted to brine to facilitate that equilibration. And we encountered a number of difficulties there, except when we decided to use diced tomatoes instead. In that case, because the equilibration is facilitated enormously, then we were able to preserve very quickly in an acidified solution, and which is what's shown here in these jars. So these two jars were our control jars where we acidified with citric acid. We also had benzoic acid as a preservative. And these jars were clear after 2 months of storage at 30 degrees C. And so these treatments were very successful. You can see the quality of the diced tomatoes that we obtained out of those jars in the picture on the bottom right. But when we did not use the right combination of preservatives and acid, then we opened the jars and there was a lot of effervescence coming out of those jars, indicating that there had been some sort of microbial activity there. And as much as I would love to put this effervescent dyes of tomatoes in my mouth in the middle of the summer, because it appeals to me as a very refreshing food, I don't know where the effervescence is coming from, so I'm going to refrain from consuming that until I better understand that treatment. The take-home message here is that we as processors need to understand very well what we are doing and make sure that our products are safe. In this slide, I'm showing you titration or acidification of a number of vegetables. So here we were looking at green leaf lettuce, we were looking at green beans, green bell peppers, and we were trying to understand what were the initial pH that were in different lots of these vegetables and how much acid we needed to add to successfully drop the pH to 3.3 for preservation purposes. And what we see here is that the initial pH oftentimes it's right there in the range between 5 and 4.5, which is critical for the germination of Clostridium botulinum. So once again, the bottom line here is that we need to be very careful and in dropping that pH quickly enough so that the vegetable is equilibrated with the brine and that we end up with a safe product for consumption that is also microbially stable. Aside from looking at acidification of a number of vegetables, we have also looked at fermentation of vegetables other than cucumbers, hoping to understand the intrinsic biodiversity. And here I'm showing you some of the organisms that we isolated from various vegetable fermentations, mostly lactic acid bacteria. We are also using the strengths of the sequencing technologies to look at biodiversity in here, and we hope to publish that within the next few months. So stay tuned if you'd like to learn more about that. The third component of this presentation, which is essentially a third research line in my program, has to do with understanding the physiology of lactic acid bacteria in plant systems. We are interested in furthering the understanding of both biodiversity and the biological functions that are present within the biodiversity. We know that lactic acid bacteria, which are the ones that dominate in vegetable fermentations, are also present in the plant system. And not just lactic acid bacteria, we have a number of gram-negative bacteria and Pseudomonas that are present in the plant system. And all of these organisms are there for a reason. They can help the plant system with specific functions related to growth hormones, availability of nitrogen, availability of phosphates, and they develop a very close relationship with the plant system. It's a mutualistic, beneficial relationship, and the plant system finds ways to maintain specialized microorganisms that help them grow. The current view is that specialized organisms that evolve to serve the needs of the plants are maintaining the seeds and transferred from one generation to the other through that seed. And so we are interested in learning more about the biodiversity that's found not only in seeds, but also in the fresh vegetables and the fermented vegetables, and investigate the functionalities that these organisms bring in the different habitats. I know that there were a number of participants in Dr. Fred Brines' presentation a couple of weeks ago that had questions about the microbiology of vegetable fermentations. So I had included the next 3 slides to touch a little bit on that. And so what I'm showing here is DNA sequencing analysis that we completed on cucumber fermentation samples that were collected at different time points, and these are commercial fermentations. Every color in this bar represents a bacterial species, and the main, and here is one tank, and the first 2 bars represent one tank, and the first and the last 2 bars represent another tank. The main take-home message is obvious here. We have a lot of colors or a lot of diversity within the first day, the first few days of the fermentation. And as lactic acid is produced and the fermentation gets started, we lose that diversity and we only keep a small fraction of the population that we initially had. So I hope this gives the audience a little bit of background on what it is that we are looking at in fermentation systems and in regards specifically to vegetables. Here I'm using classical microbiology to look at the fluctuations in the microbial population in those same fermentation tanks. And the plating data suggests that while there is an overall increase in the number of bacteria in a fermentation system up to some point, the number of gram-negative organisms, particularly Enterobacteriaceae, which represent organisms like E. coli or 157H7 and a few other gram-negatives. So the trends within that subpopulation is that it declines as the pH drops, as lactic acid is produced and the pH drops. So very important here is that it is absolutely necessary to get the pH of a fermentation dropping to minimum levels in the shortest amount of time, because that's what controls for organisms of public health significance. It's not just about Clostridium botulinum, but also about excluding undesirable microorganisms in the process. And then in terms of the chemistry, this gives you an idea of what really is going on in that fermentation. So obviously we are looking for the conversion of the simple sugars that are present in cucumbers, in this case, glucose and fructose. Most vegetables will contain glucose, fructose, and some of them will contain sucrose. So we are looking at the conversion or the disappearance of these sugars as a function of time and their conversion into lactic acid, which is shown on the top right. The production of lactic acid or the increase in lactic acid concentration. In these cases, there had been some acetic acid that was added in the brines. And so what we were looking at here was the maintenance of that initial level of acetic acid and possibly an increase if heterofermentation were to happen. So I'm hoping that this gives the audience an idea about what's going on behind the scenes in a fermentation, why it is that we ferment cucumbers or vegetables or foods in general. It's because we want to convert the glucose and the fructose, those sugars that are easily fermented by or utilized by organisms, get rid of them so in a controlled way so that we don't have spoilage occurring and we can successfully preserve the food by producing the lactic acid that then drops the pH. Why it is that we are so interested in biodiversity and biological functions? Well, the answer is simple. The more we understand about the biodiversity, the better we are equipped to deal with fermentation outcomes that are undesirable. Here, I'm showing you a project that we started to look at cases of spoilage in cucumber fermentations, where once the fermentation was completed and the pH was down to about 3.3, we were seeing increases in that pH up to 4.7. And we didn't know what was going on at all. So we started this project in collaboration with Dr. Eduardo Medina, who is currently at the Instituto de la Grasa in Seville, Spain. And we were trying to understand what were the microbial changes that were occurring that were leading to that increase in pH and the changes in the chemistry of the fermentation. So we were successfully understanding the diversity and all the microorganisms that could be potentially involved in these spoilage cases. We continue to work in this area. In fact, this is an area that is in our core project plan for the next 5 years. So there will be more to come in this subject. Understanding the biodiversity in fermentations has also helped us better understand processing defects, like I'm showing you here. This was related to blower defect, which occurs when organisms that are naturally present in fresh cucumbers can utilize the sugars that are there, and they produce a number of organic acids, but also carbon dioxide and hydrogen. Not every organism can do this, but some of those that are naturally present in cucumbers can produce carbon dioxide and hydrogen, which get accumulated inside the cucumbers, displacing the tissue and forming these hollow cavities that are undesirable in a cucumber, in a fermented cucumber. I mean, this will be a desirable attribute in Swiss cheese, where we all expect to see those holes, but not in a fermented cucumber. We want our pickles to be round and not moon-like, as it will be the case. So understanding again the biodiversity that's naturally present in this system help us identify what was happening behind the scenes. And now we are at a stage that we are using that same biodiversity to prevent this defect from happening by developing a started culture that's fast enough to prevent the activity of undesired microorganisms. The started culture is currently available, and we are hoping to transfer it for commercial use within the next year. Another goal that we have been able to accomplish by understanding biodiversity is the development of started cultures for low salt cucumber fermentations. Most fermentations are currently performed with about 6% sodium chloride, and they are not dependent on the use of a started culture to achieve a desirable outcome. However, as we reduce the amounts of sodium chloride, the use of a started culture becomes imperative. And so this new understanding is helping us identify good candidates for started cultures. We hope to have these cultures transferred to commercial production pretty soon, within the next few months as well. We are not only looking at the diversity in the bacterial community in these fermentations, but also looking at the diversity within the community of viruses that can infect bacteria and prevent a fermentation from completing. We have done this in collaboration with Dr. Jean Liu at Kennesaw State University. We currently have a very nice collection of these phages that are capable of infecting not only lactic acid bacteria, but also a number of gram-negatives. And we are looking forward to use this diversity to improve the quality, the safety, and the microbial stability of a number of vegetable products. And with that, I will start the last portion of this presentation, which is related to fermented vegetables and human health benefits. I believe Dr. Susanne Johannes-Meyer will be talking more in depth on this topic in a couple of weeks, so stay tuned for that. I am thinking that as we consider health benefits, there are a number of things we have to keep in perspective. One is that we all know that we have a microbiome that's pretty significant within us. We probably are carrying more microbes with us than the number of cells that help us exist. But, and we also know that the diversity in this microbiome is helping us to get the vitamins we need, get anti-inflammatory compounds, a number of very good positive benefits from this microbiome. Unfortunately, there is a lot of diversity within person to person in this microbiome, and that's partially due to the fact that the microbiome is not selecting for specific species, but more the selection occurs more towards functions that are needed for a specific need. So within that parameter, then we need to understand that there is a diversity that may or may not be needed. There is a diversity in fermented vegetables that may or may not be needed in the gut. And we need to make those relationships. We need to start making those, start deriving the knowledge that's needed to understand those interactions. In considering those aspects, I'd like to also highlight the fact that the fermentation that happens in a cucumber tank or vegetable fermentation in general is focused in the conversion of simple sugars, glucose and fructose, to lactic acid, which is not the case in the gut, for example. The energy sources that arrived to the gut are mostly those compounds that cannot be digested. So what we really end up with in the gut is dietary fibers in the case of fermented vegetables, and all those compounds that we cannot digest. So we have to keep that in mind, because the fermentations that we typically see in a vegetable then is different from the fermentation that is to occur in the gut. The substrates are different, and although some of the functions may be similar, not all of the functions are compatible. So I think we as researchers need to keep that in mind and make those considerations as we make progress in developing fundamental knowledge along health benefits. There are three scenarios that I can see in better understanding health benefits from fermented vegetables. And one is assuming that the entire biodiversity that's present in these fermented vegetables is somehow interacting with the microbiome beneficially, hopefully, and we have to understand all those interactions. And I am very thankful to you, Ben, and other researchers who are making very good progress in this area, using the complexity of the sequencing technology and bioinformatics to really understand the interactions that occur here, biological interactions, chemical interactions, and so forth. Alternatively, we also have the opportunity to simplify this concept and look at an indigenous organism in vegetable fermentation, one that predominates, and then study the role of this microorganism in the gut. And many researchers have looked at potential benefits that can be derived from fermentation, fermentative organisms such as Lactobacillus plantarum. And I look forward to learning more from what my peer researchers are doing in that area. The third scenario then is to adopt probiotic cultures, adopt cultures that are known to cause a beneficial effect, and incorporate them into vegetable fermentations to understand how it is that they are interacting with the fermented vegetables, so that we can better understand what it is that is being delivered to the gut. In this case, when we use exogenous or alloctonous cultures, then we have oftentimes the ability to specifically track this probiotic culture. And so that's where we decided to start along this line of research, where we took a number of alloctonous probiotic cultures, incorporated them into refrigerated cucumber products, thinking that the refrigeration will be the best tool in maintaining long-term viability of the probiotic cultures. And we were excited about this. We got lyophilized probiotic cultures. We added them to refrigerated cucumbers. The quality of these cucumbers was excellent and preferred by consumers. However, these organisms died off in a matter of 4 days. So we couldn't keep these organisms viable at high enough level that so that they could exert a beneficial health effect for the consumer for long enough. Then we decided to look at what other things can we do to prolong that survival. And what I'm showing you here is that by either growing the probiotics in the product for a certain period of time, or by supplementing with arginine, then we get the probiotic to survive at high enough level for 2 months. Now, 2 months is still short in terms of the desirable shelf life for this type of product. We need to continue to make progress along these lines. But it's a fascinating discovery that by doing these 2 steps, incorporating these 2 steps in the process, we can induce aggregation by the probiotics and long-term survival. So I'm very excited for the opportunity to share all of this information with the audience and with you, Ben, today. And with that, I'll be happy to take questions.
Thank you so much. There's so many exciting things that you're working on. And again, we're so grateful for your work because it's really helping the industry. I have 2 quick questions I'm going to ask, and then we'll maybe check in with the participants to see if they have additional questions. One question is, as a member of the Fermentation Association's advisory board, one thing we often talk about is industry needs. What are the big industry needs? And I often, as a scientist, think about, what are the science needs? So what would you argue, of all the things you're working on, or maybe other things for pickling and fermentation, what's the biggest gap in our knowledge scientifically that we really need to be working on to better understand these products?
I honestly think there is still a lot to do in regards to the richness of the biological functions that are present in these systems. Every vegetable is different. Every matrix, the pectin composition is different within vegetables. Sugar content is different. We still need to, we have a wealth of functions there, a wealth of chemical composition that we don't still fully understand. And it will be fantastic, I think. It will be fantastic to be able to comprehensively understand what's really there and how we can use it for the benefit of not only processing, but also for the benefit of human health. So I think that's the biggest void. A lot of resources needed there.
Another question that came in, sort of a theme that came across questions that were submitted before this session, was about scaling. So, some of the work that you talked about were in these really big, amazing tanks that are outside. And then you also talked about sort of smaller scale fermentation for the issue of food waste and surplus. So what do you see as some of the challenges of scaling what you learn at the large scale down to these smaller scale fermentations? Does everything transfer, or are there things that don't really work across those 2 different scales?
Yeah, that's a very good question. And the greatest challenge we've come across is the availability of oxygen within the different systems. Also, when we are working in jars and we are carefully putting vegetables in a jar, that's the best scenario. But in commercial operations, the tanks themselves are so tall that we end up throwing the vegetables in there. So we have to cushion for the fall, prevent damage as they fall in these tanks. We also, another complication is that with giant tanks, you can't really flip them to get a good mix. So you have to use either purging systems or circulation systems to be able to maintain that homogeneity in the system. So it's challenges of that nature on the technicalities of how to do the fermentation. Now, the fermentation itself, the chemical conversion is oftentimes the same. What can impact the fermentation is factors like temperature, the availability of oxygen, the amount, the coloration. It becomes tricky to have the right concentrations of salt, the right concentrations of acid and preservatives and whatnot. So, so those are the areas where we have seen the most challenges so far.
I might sneak in one more question. So
Sure.
I think all of us think about sodium chloride or salt, table salt, as like essential for fermentation, right? It's the tool that we use to do fermentation. And it seems like your work, you're sort of taking that, you're sort of pulling the rug out from underneath us and saying what happens when we remove that essential tool. So what microbiologically, what happens when you start taking, reducing salt levels and supplementing other types of salt in there?
Yeah, very good question. The first time I heard we were going to have to develop a sodium chloride-free fermentation, I was like, oh my gosh, that's not a fermentation. What are you talking about? But what I've learned a couple of things since then. One is that the indigenous microbiota is always there. The salt is modulating the activity within this population, and it tends to favor the lactic acid bacteria. But what I've learned is that it is not the main factor modulating that microbiota. I'm thinking that pH and the production of that lactic acid and or acetic acid is the main factor in really excluding the non-desirable microorganisms and favoring the fermentative microbiota. So in that sense, I think that's why we've been able to create a system without sodium chloride. However, I must highlight that when we use the systems without sodium chloride, then we are also having to incorporate preservatives that then help us control for the undesirable changes that occur. It is microbiologically speaking, it is a more vulnerable system, the ones without the sodium chloride. So we still have a lot to learn along those lines.
Great.
Yeah, thank you for the question. Yes.
Yeah, no, I think your work is so important. And I'll just point out that in the future, we're going to be having some more webinars about the microbiology of fermentation. I'll be doing a deep dive into some of the work we've been doing in my lab, and we'll also have some other really cool microbial deep dives. So definitely stay tuned for that.
Okay.
I'm going to pass it over to Amelia.
Yes, thank you so much, Ilenys. That was a fascinating presentation. I have a few questions from our audience now. Okay, so first we have, what about mixed vegetable fermentation? Have you done any, has the lab done any research on that, like a cabbage plus cucumber to make a hybrid pickle sauerkraut product?
Yeah, that's a very good question. I'm dying to get there, but we are not there yet. I wasn't brave enough to start with mixed vegetable fermentations because even though vegetable fermentation, it's a very old process. People have done it for centuries around the world. It's a very well utilized system. But when you look in the scientific literature, there is very little knowledge documented as to what are the changes that occur in these fermentations in terms of the microbiology and the chemistry. There is limited knowledge documented. So what I've decided to do is start simple and understand what happens in fermentations with individual vegetables first, and then start thinking about, what makes sense to mix. There is a diversity of pH values among these vegetables. The buffer capacity is different. Sugar content is different. The matrix itself are different. So I want to learn, take a more systematic approach and learn about these fermentations individually, and then hopefully gain enough knowledge to then say, okay, it makes sense to combine these two fermentations from the chemistry or the biology point of view. Let's see what happens. And so that's where we are at the moment, but it's a very good question. I hope we get there someday.
Yeah. So should different pickling methods be followed for different kind of vegetables?
I think based on the data we have as of today, I think there is a need to modulate the composition of the fermentation cover brines, because every vegetable has different needs. So I think there will be the need to tailor and customize the ingredients that go into the cover brines. So yes.
Good. All right. Can you say anything about like the health benefits of potential health benefits of a live microbes versus dead microbes?
Okay.
Yeah. What are the health benefits there?
Yes, I know that there was I want to say about 15 years ago there was a movement within the scientific community proponing dead microorganisms as sources of probiotic effects, even without the viability. That was the thought there. I'm not sure how far the research went along those lines. I have the impression that to some extent, these dead organisms could trigger some sort of responses from the gut, because they, the composition of their membranes can be recognized in the gut by certain molecules there. So there might be some potential there, but I don't know where the science, I honestly haven't kept up with the science of that area. However, I want to highlight there are a couple of things that are important when thinking about health benefits. Is that very unique to vegetables is the fact that there is dietary fibers in them, the pectins and so forth, that are consumed together with the organisms that are present in the fermentation. And that combination of organisms that are pre-adapted to an acidic pH and pre-adapted to these pectins can be very powerful in the gut. I think we need to better understand those interactions, and I think we'll be pleasantly surprised about what we find there. So
I loved your thoughts on reducing food waste and what the USDA is developing to help people ferment from home or from restaurants. How critical do you think fermentation is to reducing food waste to help our modern food system adapt and be more sustainable?
Yes, I think the critical part of fermentation and or acidification is the fact that these processes are very simple and yet very effective in controlling and removing for undesirable microorganisms. I think that's what we can exploit in reclaiming surplus vegetables and defective vegetables. These fermentations are, if applied properly, they are very powerful in eliminating the organisms that are not desirable or organisms that are of public health significance. So a lot of potential there. Of course, there are other possibilities for surplus vegetables and defective vegetables. I'm sure that the scientific community will come with a number of opportunities there. But I think fermentation will truly be an important component as we move forward towards that new era. Yeah.
Well, Ilenys and Ben, thank you so much for joining us today. We didn't get to everybody's questions. I am going to put in the chat right now another copy of Ilenys's email address. She's generously offered she can continue taking questions. Thank you all to you so much out in the audience for attending today's webinar. We will be posting a full recording of it on TFA's website in the next 24 hours, along with a copy of Dr. Pérez-Díaz's presentation. We also have a number of great webinars coming up in the next few weeks, including the presentation mentioned earlier, the third in our USDA ARS series. Dr. Suzanne Johanningsmeyer will talk with TFA Advisory Board member Dr. Maria Marco about prospects for health promoting pickled vegetables. Note that we had to change this webinar, so there is the date of it. There's a date change. It will now be available 2 days earlier on Monday, September 21st at 10 a.m. Pacific, 1 p.m. Eastern. We also have scheduled sessions coming up in the next 2 months with Sandor Katz, Mara King, Charlie Kalish, Michael Ganzel, and Kirsten and Christopher Shockey. please go to fermentationassociation.org to check these out and to register. And while you're there, please subscribe to our YouTube channel so you can get a notification when these videos go live. Again, thank you so much, Ilenys and Ben. We appreciate your wonderful insight.
My pleasure. Thank you for the invitation.
Great to be here.
All right.
Bye-bye.
Thanks, everyone. Bye.